webinar of our new water testing series. My name is Aletia Cruz, and I'm one of the microbiology product managers at Conda. So for our first timers, Conda Lab Talks refers to the webinars we will be preparing for you from time to time on subjects of interest in different microbiology areas. The purpose of this Conda Lab Talk, as you know, is to discuss Legionella, which is a real concern around the world. So I would like to mention, because maybe some of you are new, that this platform has a question box. So please send us any questions or doubts that you may have during the presentation, and we will answer these at the end of the webinar. So we will begin with the bacteria and the diseases this can cause. Then we will review the reference method, followed by an alternative method, an overview of the combined methods, and we will end with a quick Q&A. So let's dive right into the bacteria and the disease. What is Legionella? These are aerobic gram-negative bacteria that are found predominantly in water sources and freshwater environments worldwide. However, it can also be found in potting soil. In both environments, it survives as an intracellular parasite of free-living protozoa. It can multiply between 20 and 55 degrees Celsius with an optimal growth temperature of 35 degrees Celsius. Temperature is a very relevant factor to take into account for its prevalence in the air conditioning system. So the first strains were isolated in 1943, but the genus was established in 1979 after a large outbreak of pneumonia among the members of the American Legion. The genus Legionella has over 60 confirmed species and more than 100 assumed from NGS data. Also, there are currently 7 distinct serogroups. Previously, some investigators had proposed dividing it into 3 separate genera, Legionella, Fluobacter, and Taglochia. Nevertheless, recent studies do not support the division of the family Legionellaceae and confirm it as a single group. I would like to mention as well its capacity to survive within biofilms in building water systems, since these structures are able to provide shelter and complex nutrients. Still, some studies have shown that amoeba may be required in the biofilm in order for Legionella to multiply. More investigations, of course, are required, but controlling the biofilm associated Legionella could provide the most effective control measure to prevent legionellosis. Legionellosis commonly has 2 clinical presentations. One, which is the most known, the Legionnaires disease, a multi-system disease involving pneumonia, and a milder form that is a flu-like illness, which is the Pontiac fever. Clinically, it is not possible to distinguish Legionnaires disease from other types of pneumonia since its symptomatology is common. Fever, non-productive cough, headache, etc. And before rapid tests to rule out COVID-19 tests for Legionella and Enterococci were performed as well. So to correctly give a diagnosis, microbiologic testing is key, which we will discuss in a bit. Legionella pneumophila is mostly responsible of all reported cases in the US. These data may be inflated because most diagnostic tests are specific for this species. Currently, there are 15 serogroups of Legionella pneumophila, but almost 80% of all cultured confirmed or antigen confirmed cases are caused by one serogroup, which is 1. Serogroup 1. Approximately 30 other species of Legionella have been associated with human disease, and as we discussed previously, we could presume that all Legionella are capable of intracellular growth in a host cell, and therefore likely that most species can cause human disease under some conditions. Infections due to less common strains of Legionella are infrequent, mostly due to their rarity and also the lack of diagnostic reagents. The transmission of the bacteria is via aerosol to human host that is susceptible to infection, being the risk group predominantly males over 50 years old. In the European Union, according to the ECDC, which is the European Center for Disease and Prevention and Control, there has been a 23% increase in cases in just a year. Four countries, sorry, 4 countries account for 71% of all notified cases. And it is because of this trend that an analysis of Legionella in homes has been proposed, although it is not yet contemplated in the legislation or near it. It can have up to 15% lethality, although in the EU it has around an 8%. Nevertheless, legionellosis remains an important cause of preventable mortality in Europe, at least. Therefore, the importance of regular checks and control measures applied to water systems. Okay, so a person is telling me that there is no sound. I don't know if the rest can hear me correctly. I'm gonna guess yes, you can hear me correctly because it's just been one person that is telling me that it's okay. Okay, so maybe it's just one person that is having problems. So I'm gonna continue. Thank you for the rest. Okay, as I was saying, the nosocomial outbreaks and prevention. It's important to have regular checks and control measures applied to water systems, especially in hospitals and long-term healthcare facilities, and in any setting where high-risk population may be exposed. So in this timeline, we have some of the outbreaks in Europe where the mortality is not that high. For example, here in Murcia, Spain, of almost 500 cases, only 6 patients died. But however, in France, a few years later, with 86 confirmed cases, there were 18 fatalities. It is worrying the fact that there is no indication of decreasing burden, and there have been sharp increases in the overall EU notification rates in recent years, as we can observe in this chart on the left. Also, we can take special notice in July, where the sharp increases occur, and this is constant through the years and consistent with the opening of recreational spaces such as public swimming pools, water parks, and also the use of air conditioning. It sure highlights the clinical importance of Legionella and its control. So therefore, now we will review in detail the reference method for Legionella, which is ISO method 11731 with the 2017 update. Those who have experience or know the method will know very well that it is complex and honestly a bit tedious, but it is necessary to ensure the reproducibility of the method and the results obtained, no matter where they are performed. So we can look at it as a series of matrices with which you will make a decision to carry out one method or the other. This depends mainly on the sample to be analyzed and its origin, which will allow us to ensure the detection of Legionella. It is very important since Legionella is a slow-growing bacteria and the accompanying flora can prevent or even delay its development. There are different procedures according to the sample to be analyzed, and this is to ensure the detection of the Legionella in it. The origin and nature of the sample indicates the treatment to be followed. So let's go right at it. In the first case, we have a sample with a high concentration of Legionella and a low load of accompanying microorganisms. This is the most simple, where the sample is just stripped directly onto Legionella BCYE agar and BCYE plus antibiotics agar. So next, this is where it starts to get complicated. We have a sample with a low accompanying Legionella and a low concentration of Legionella and also a low concentration of accompanying microorganisms. We can approach this two ways. The first, we have by membrane filtration with either an untreated sample and directly placing it on a BCYE agar, or the second option, okay, here it is, also by membrane filtration following with an elution to concentrate it, and you have to divide this into 3 portions and apply a pretreatment. Here it's optional and here it is mandatory. You have to apply a pretreatment to one part and heat treatment, to another an acid treatment, and you leave the third one untreated. Okay, and then you place this filter on the one of these agars, BCYE agar, and you have to use another one of these 3. That is up to you. Usually, when the ISO gives you options of some agars to use, the most common one is the GBPC agar. So next, we have number 3, which is a high concentration of accompanying microorganisms, and you just don't know the concentration of Legionella. So here, the sample is divided into 3 subsamples: direct, which is untreated, concentrated, and diluted. Next, to each one of these samples, you have to do a pretreatment, which would be acid pretreatment, heat pretreatment, and then the last to leave untreated. So now here it's complicated because we have several different subsamples and pretreatments to do to one and another. So after you've done all this, each one of these is stripped onto a GBPC agar. And the last one is, we're almost done, so stay with me. The last scenario is when you have a sample with an extreme high concentration of accompanying microorganisms. So here, the sample is first subjected to a combined pretreatment, which would be first the heat pretreatment and then the acid treatment. Then you divide it again into 3 subsamples, into 2, direct and diluted. And these 2 are stripped onto the GBPC agar. Okay, now we've reviewed all the possible scenarios you may have, and the common procedure to all of these is to incubate at 36 degrees Celsius for 7 to 10 days. There is no difference between the counting done at day 7 and day 10. However, if you are working with natural samples that have wild strains of Legionella, this may require the full 10 days incubation to develop growth. The presumptive confirmation must be done by subcultivating onto BCYE agar and BCYE without cysteine agar for 2 to 5 days. The results. Okay, if you have a positive growth in BCYE agar and a negative one in BCYE without cysteine agar, the colonies are considered to be Legionella. Okay, so now to visualize a bit the results, what you would observe in the first image, we have a BCYE plate with an overflowing growth of non-Legionella microorganisms. There it is almost impossible to see Legionella in this plate, and this may be because the disinfection was not appropriate. So in contrast, here on the right, we have another BCYE plate grown with two Legionella pneumophila bacterial colonies, the ones with the pointing arrowheads, which are this one in the upper left and this one on the right, amongst other bacterial colonies that are no interest to us. In this last image at the bottom, we can appreciate Legionella colonies after 3 days and 7 days of incubation. The younger colonies have a characteristic cut glass appearance and iridescence of younger that disappears after a long incubation, as you can see here. The texture is not the same; it is lost. So, as you may have noticed, the ISA method is complex, and it has several sources of variability, be dilutions, pretreatments, or different media. So in summary, the bottlenecks are time and laboratory work, as sometimes the results can be stretched up to 20 days, and also because there must be a continuous inspection of the plates almost daily. Also, some Legionella cannot be grown on this media and are referred to as Legionella-like amoebal pathogens, which is rare. But finally, also the confirmation of the presumed colonies is mandatory with its a couple or several days of incubation. So, in response, currently there are available alternative methods that are quicker to give us a presumptive result. There are several techniques, but today we will talk about one in specific, which is real-time PCR or qPCR. Okay, let's talk briefly about PCR that I'm sure most of us have heard about, especially this year. PCR is a polymerase chain reaction, as its name indicates, where copies from a single DNA fragment present in a sample are obtained through amplification. Each PCR cycle has different temperatures and stages. First, it's the denaturation step, which is the separation of the strands. Then the annealing stage, where primers and polymerases incorporate to elongate in the target strand in the extension step. The difference between a traditional PCR and a qPCR is that fluorescent labeling enables the collection of data as the PCR progresses. There are several techniques to do so. Some are stains, but the most specific one is the in-probe-based qPCR. Probes are designed DNA fragments that are complementary to the target DNA. In this DNA probe, there are two molecules, a fluorophore and a quencher. The latter prevents the emission of the first when together, but with the polymerase action, the quencher is released and thus allows the fluorophore to emit the fluorescence that will be detected by the equipment in real time. In this image, okay, now it appears, we see a typical qPCR curve where the different colors would correspond, for example, to different bacterial species. So, to compare a bit, let's review each method. Okay, let's review each method. Um, in both, we can see that we have common processes such as sample collection, but with PCR, we go directly to the filtration and then the DNA extraction, consequently the PCR, which overall is very fast and the results would be obtained the next day. It is also very important to notice this is a quantitative method, not just qualitative of presence or absence. And in the reference method, as we've seen, there are multiple steps and it is quite a lengthy process. So each method has its advantages and limitations. So we will see a very interesting joint use of these both. To begin, we have the reference method, which is the one referred to in the legislation. So that's very important. It's the easiest one to implement. It requires a confirmation of results and therefore gives us a high reliability of results. The protocol is complex, but it is also a very normal routine in any microbiology laboratory. So no expertise, staff, or equipment is required. On the other hand, we have a method that is quicker, obviously, with a high sensitivity and specificity. There are specialty equipment required, but the protocol should be simple enough to follow. Also, there are some viability PCR available that include a reagent to block any free DNA in the sample, meaning any dead Legionella that wouldn't grow in a culture won't be amplified, thus avoiding false positives. A combined approach has been then proposed by Paloma Belver in Galicia's Public Health Laboratory to improve the lab work because due to the low frequency of positives, interest arose in using PCR as an initial screening. Is the sample associated with a clinical case or outbreak? No, then you can continue with the ISO method without any time pressure. However, if you have a sample associated to a clinical case or that it's important, it has a health importance, you can proceed with a PCR. The result you obtain is negative. Okay, so there is nothing more to do. However, if you have a positive PCR, a confirmation culture must be performed but the days to give a result have been reduced considerably. Okay, so to better review this strategy, let's assume we have, in this case, a PCR result that is higher than the allowed level, that would be around 4 times 10 to the third power genomic units. So um, the PCR and the culture are repeated for the same sample. And then we have matrices of decisions depending of the results obtained. Here we have, if both methods give us a negative result, okay, then there's nothing else to do. If we have a positive result, no matter if the PCR is positive or negative, we have to follow the guidelines established for the level detected by, and this is in the legislation. So the third case, we have a negative culture, but the PCR is positive. So we have to do some more investigation because something may be wrong in the procedure. Maybe further sampling is required or other variables that may have interfered in the result. And so to summarize, the PCR allows us to do a previous screening and confirm it always obligatory with the culture. We cannot leave the culture out of our routine. It is mandatory to do so, even just if it's for the confirmation or of a positive result obtained. So the result of this joint strategy that has been implemented by Paloma in Galicia's lab is that's shown in this chart. Let me see if I can. Okay, so okay, perfect. It is working. The pointer. So let's review. It is a bit of a not so easy to read chart, but let's just focus on these first 4 bars so we can know what we're talking about. In a darker blue here, we have the numbers of PCR performed. So about almost 100, let's say, number of tests. In orange, we have only the positive PCRs of these 100 number of almost 100 number of tests. We have less than 50 positives. Okay, perfect. These then must be confirmed by the culture. So in yellow, we have the positives of the positive PCRs, meaning of the almost 100 samples, we have avoided 53 cultures. 53 culture procedures were avoided by using this joint approach. It is also worth noting that for Legionella testing, it has been estimated that for a single sample, just one sample, around 30 plates of media are required. So imagine if you could avoid 53 cultures of what. Okay, so besides all the lab work, because the antibiotics are included in the media, the disposal is also quite difficult. So there we have, with this joint strategy, a huge load could be avoided. It gives us versatility and, most importantly, speed of response to emergency situations. So we are almost done. At Conda, we have the available products for your ISOM protocol with the same base, and depending on the added supplements, BC, YE, JVPC, or MWY agar can be prepared. Also, we have plates ready to use media, so you can quickly begin your procedure without any delays. And regarding other Legionella products, well, you just have to wait for our new launches that are coming soon in 2021. So with this, we conclude the webinar. And we will have some minutes for the Q&A part. So let's get right to it. Okay. Just a second. Okay, so the first question, are there other alternative methods for Legionella? Okay, so of course, it's not only PCR, there are immunological detection methods. There are several tests on the market with different antibody markings, and the reading of the results also varies. It can be colorimetric, cytometry, etc. It can be also combined with other techniques such as magnetic separation. There is also flow cytometry and mass spectrometry. Actually, MALDI-TOF is implanted in the Netherlands because it is very fast and has a high reliability. I think also it has been tested in Germany for drinking water and some other countries. However, it requires a significant investment in equipment. Staff must be also experienced with this equipment and technique, and the growing samples are required to do the analysis. You just can't use sample that you took from a water bank or something like that. You have to have growths of Legionella. The problem with all of these is that, again, these are not standardized methods. And in some of the cases, the specificity depends on the antibody used, and also there are many interferences and sources of variability. And all the positive results must be confirmed by culture, as we've mentioned. Okay, yeah. For what is the heat and acid treatment that the ISO refers to? Okay, so for the heat treatment, you have your sample and you add it to a sterile container, of course, and you place it in a water bath for half an hour. I believe it has to reach 50 degrees Celsius or something like that. It is advisable to use small volumes, so you can concentrate it or not, depending, and to guarantee a very short time of period until the desired temperature is reached. It is important, as always, to not overheat the sample if using thick recipients. So it is advised to cool quickly to avoid any overheating, that because it can damage the Legionella. On the other hand, the acid treatment consists of diluting the sample with an acid solution and mixing. It is very important to consider the dilution factor at the end for your results. Or also, the acidic solution can be directly transferred onto the membrane filter, and then the solution is filtered up. It is very important that the diluent does not wash the surface of the funnel that was not in contact with the acid solution. So you have to be very careful. And oh my God, we have almost uh, out of time, but let's do another question. Are the PCR kits accredited? Okay, so it depends on the kit you use, but there's an ISO for Legionella PCR, and if this kit is according to the ISO method, it is not necessary for it to be accredited. So it just depends, obviously, on the brand you are using. So okay, I see that some of you are asking for the presentation. As you know, after the webinar, you will have the recording being sent to you, so you can watch it again. If you want to take a good look at the ISO method, I know that slide was a bit complex. Let's look at it a bit quickly again. It is a bit confusing, it can be a bit complex. So yeah, you will receive the recording of the presentation so you can look at it with more time and more detail. And I see we have other questions, but we are out of time. As you know, we will answer all of your questions by email. Do not worry, these are all registered. So lastly, we want to invite you to our next webinar, which will be in December, and we will talk about enteric oxide. So again, for our water testing series. The communication will be sent later. You will receive it, so please subscribe so you can attend. And we hope that the seminar was interesting and that we have resolved any questions about Legionella and ISO 11731. And well, until next time, thank you all.